Introduction. Now, Merje is from Estonia. Her PhD is in plant pathology, actually in rusts, and she also has done postdoctoral work studying rusts. She tells me they are her favourite. Now she works for the Ministry of Primary Industries. She is based here in Auckland, and she was involved in the initial MPI-led response to Myrtle Rust. Passing you over now to Merje.
Tēnā koutou. Thank you, Renee, for the introduction and also for the invitation to present in this webinar series today. I would like to start by saying that while I'm the one presenting here today, it is on behalf of a number of people at PHL who are working in the Myrtle Rust response for more than a year.
Merje, I don't think we can see your screen yet, so you just need to accept the screen coming to you. You got your two monitors going there. This is what happens. There can be some. There we go. Looking good. Looking good as you were.
Cool. So yeah, let's just continue. Okay. There. I'm sure that Ostropachydia obsidii does not really need an introduction to the people attending this webinar today. And also, if you've seen the previous talks in this series, they have given some good overview of the pathogen and the disease. But there are a few things I would like to emphasize about Myrtle Rust, which is the extremely wide host range, over 450 known hosts and counting. The characteristic rust symptoms, which are caused by the uridinial stage, its ability to spread by airborne spores, and the fact that once it's established, the fungus is extremely difficult to eradicate. Before I get to the Myrtle Rust response in New Zealand, I would like to briefly cover what we do at PHL, at the Plant Health and Environment Laboratory of the Ministry for Primary Industries. The lab has 4 teams. There's the Mycology and Bacteriology team that I'm part of, the entomology team, which is split between the Auckland and Christchurch sites, the virology team, and the botany and post-entry quarantine team. So we currently have over 60 people working across 2 different sites. As the New Zealand's National Plant Health Laboratory, we are responsible for identifying and validating suspected new pests and diseases. And this could be by identifying unwanted pests at our borders or ensuring that new finds from New Zealand are accurately identified and investigated as needed. In addition to diagnostics, though, we also provide scientific advice and support to other MPI teams, and to ensure that we are using the most efficient and fit for purpose methodology. We also are working on diagnostic capability projects. This is to explore and adopt new tools and technology constantly. We also collaborate with our international colleagues at many levels. So we are part of IPPC, Quads groups, SFED. We are taking part of Fresco projects. So a lot is going on in that capability and readiness field. And all our work is taking place in a high quality environment, as PHL is accredited to the ISO 17025 standard. The role in the Myrtle Rust response, we had a number of responsibilities. So we obviously provided the diagnostics of the pathogen and the host, but we also managed the notifications from surveillance teams and the public, provided scientific advice, and did some trainings for survey teams and others as required, whether it was about symptom recognition of rust or sample collection or imaging. So we did quite a bit of things. Before I start talking about the response, I would like to take a step back and briefly talk about some activities that happened before Myrtle Rust was found in New Zealand. At PHL, we have several preparedness activities, and Myrtle Rust had been on our team's radar already before it was detected in Australia. So before 2010, the lab had adopted the nested PCR, which at the time was the best diagnostic assay available. But once Ostropoccinia absirei was also found in Australia, additional diagnostic preparedness activities were also initiated at MPI. So these included a project to develop a probe-based real-time PCR assay for Ostropoccinia absirei. At the time, you probably know it was just Poccinia absirei. And there was also a project for preparing a barcode database for Myrtaceae plants to be able to quickly and accurately identify the hosts. The sensitive and specific real-time PCR assay was validated with imported DNA. So we had specimens from Australia, Brazil, Hawaii, and New Caledonia. And it was also tested in New Zealand and Australia. So it was shown to be a very robust assay. And it was published in 2014. I've included the reference there. In 2018, the test was also included in the IPPC diagnostic protocol for Myrtle Rust. So that's a great recognition of the assay. And the Myrtaceae barcode database, it was published in 2015, and it covers over 100 species of plants in the Myrtle family. The work was outsourced to Science, so they were the one generating all the data, and I've also included the reference here. A special focus in this project was placed on species with known susceptibility to Myrtle Rust, or on hosts that are of cultural or economic significance in New Zealand. And in addition, also, we had a project to develop a lateral flow device for in-field Myrtle Rust diagnostics. But since the sensitivity of that assay was not as high as expected or as we would have liked, this tool was not fully developed or used in the Myrtle Rust response. But yeah, this is just to say that there were numerous activities taken by the lab. And also we provided advice to the other teams on preparing the response plan or reviewing plant import health standards. So yeah, there were lots of stuff happening. So when the suspect Myrtle Rust found from Raoul Island was first called through in February 2017, our team was ready to provide fast and sensitive diagnostics to support the surveillance and the response teams. So from Raoul, originally a small number of samples was sent. They were all securely packaged and received here after several days of transit. We were able to quickly confirm it as Ostropoccinia absirei on the Metrosideros spermaticensis plants. It just took us a day, or probably even a little bit less. And when the first mainland find was reported from Kerikeri in May 2017, the team was already geared up and ready to respond to the find. Except this time, we needed to handle much more fresh plant material with freshly sporulating Uridinea, which required a little bit extra precautionary measures to ensure that the pathogen was well contained and not spread by sampling teams or during transit. In the lab, in our PC2 facility, the team created a bio bubble around the biosafety cabinet to ensure an extra level of containment in the area where the packages were opened and samples were processed. The material was also double bagged, sometimes triple bagged, wiped down with decontaminants in the field while packing and before shipping it to us. Really well sealed, well labeled. And these were then opened in the biosafety cabinet within the biobubble to process the sample and proceed with identification. And the diagnostics itself contained the following steps. So we had the examination of the symptoms first, then if needed, based on photos, we requested sample to be collected by survey teams. We examined the micromorphology of the rust spores in the lab, extracted DNA to confirm the pathogen as Ostropaccinia psirrii with our specific real-time PCR assay. And then if it was suspected to be a new host, also the host was identified with DNA sequencing with this barcoding, with the barcodes that were developed in that project. And that was done by Cyan, by the way. So this was the process that happened for the first 4 months. So all the finds were confirmed with morphology as well as PCR. But since processing fresh infected plant material in the lab and completing the morphological identification was extremely time consuming due to the additional measures that needed to be taken, the team decided to make some changes in the diagnostic process. And here is what was done. So instead of collecting whole leaves or bigger parts of the plant and sending them to the lab, the survey teams were sent vials which contained metal beads and CETA buffer, and they were trained to collect samples for us in the field. So infected and healthy host material was cut into small pieces, placed in a buffer. So once those tubes arrived in the lab, they could be processed directly for DNA extraction without opening up the tubes or risking the spread of the spores. This meant minimized risk of containment breach or spread of the spores and faster processing as well. Starting from September 2017, when Myrtle Rust was confirmed from multiple distant locations in New Zealand, eradication was determined unfeasible. We also made some changes in the diagnostic process. The main change being that image-based diagnostic was determined to be enough. There was also in consultation with what Australia was doing. And so we only received images and only requested samples if there was a suspected new area or new host. So those we still confirmed with PCR, and we still do today. Over the 14-month period, we confirmed 1,218 positive Myrtle Rust finds. In the second part of this presentation, I would like to focus on the notifications about suspected Myrtle Rust finds. These would come through either surveillance teams. They were specifically trained to complete Myrtle Rust surveillance or from the public. The majority of the survey teams were coordinated by Assure Quality, but also DOC and the MPI's high-risk site surveillance teams were on the lookout for Myrtle Rust. And the public was calling our 0800 number or used the Myrtle Rust Reporter app that was developed. To keep the public informed and engaged, MPI also ran large public awareness campaigns to remind everyone what the fungus looks like and what to do if you find it. So you might have seen ads like this emphasizing the message not to touch the infected plants and asking to report the finds. Or maybe you saw the loud messaging like Myrtle Rust could explode this spring, or perhaps you saw the video clip. I'm sure that the majority of the people in New Zealand learned in 2017 what Myrtle Rust is. So as already mentioned, PHL also managed the notifications, and these were triaged at the call center before they were sent to us. We completed the image-based assessment first, and then requested additional images or samples if they were required for confirmation. There were days where we would examine hundreds of images, and the three of us from our team who were completing the image-based diagnostics saw a lot of Myrtle Rust. Oh yeah, sorry, I already mentioned that we also requested samples if needed. And as part of our role in the response, we also completed daily reporting of the findings to the response team. So that is all part of the Coordinated Incident Management System. For short, it's called CIMS. This is a response model used in all responses in New Zealand, including biosecurity responses. You might be familiar with the daily COVID-19 situation updates from our response early this year. So this was also pretty much what happened during Myrtle Rust response, except it was obviously much less visible to the ones who were not directly involved. But the system is all the same, so there's lots of working together and making sure everyone is on the same page. Here you can see some data about the notifications. In the beginning of the response, a large number of calls were received by the call center. You can see that it's nearly 1,400 in May. Much smaller number was passed on to the lab. That would be the brown line. So that was a result of the triage system that I mentioned before. It was set up to rule out the cases that were clearly not Myrtle Rust. And this helped to make it easier to manage the high number of calls at the lab, as all images needed to be really closely examined by people with experience of recognizing rust fungi symptoms. Here are some examples of the images that were sent through and could be ruled out as not being Myrtle Rust. Up on the left, you can see rust, but it's non-Mortaceae host, so it wouldn't be Myrtle Rust. Range of other kind of leaf spots or yellowing, and even some pretty yellow lichens that caught people's attention. So people certainly were paying much more attention to plants around them, and calling through if ever they thought they had found something unusual. And some examples also of cases where we needed to take a closer look to determine what had caused the symptoms. So these are the kind of photos that would come in addition to the clear Myrtle Rust infection images. Some of them turned out to be insect damage or insects on the leaves, actually. For example, the one on the up left side. The one in the middle actually turned out to be pollen. And there were some cases where we needed to get the samples in to the lab to just confirm if it could be Myrtle Rust, but just not yet a stage where the Uridine spores were formed. So in that case, we would request a sample and do some PCR testing. Going back to the notifications, over the 14-month period, we received over 7,000 notifications at the call center. Of these, 3,764 were passed on to the lab. And as I mentioned before, just over 1,200 of these were confirmed as positive for Myrtle Rust. You can also see some interesting peaks in the number of calls coming into the call center. That is the orange arrows. And this was particularly from the public, not so much from the survey teams. And these were directly linked to the public awareness campaigns that we were running at MPI. They happened in May, late August, and in November. So that really shows the importance of reminding people to watch out for Myrtle Rust. And engaging different groups into searching for pathogens and pests was also beautifully demonstrated in the Myrtle Rust response. The trained survey teams were reporting most of the hosts, of the new hosts from New Zealand, but it was the public who found the vast majority of the new distribution areas. So I would really like to emphasize the significant role of all the people who were out there looking for Myrtle Rust. So everyone had a significant impact on finding where it was. We confirmed 30 hosts in New Zealand. This also includes some hybrids that were identified. And here are just some images of the symptoms on a few hosts. Some of these were significant finds, like infection of Metrosideros bartlettii or Syzygium myrae, which are both vulnerable hosts in New Zealand, or the find from Kejoa, which was the first confirmed infection in a natural setting. As you can see, we received some really nice images. These are all from the survey team. While image quality was often an issue, and we had to request additional photos to be able to see what is on the leaves, to zoom out to have good enough quality, the quality did improve significantly during the response, and the teams really quickly responded to our feedback, and that really facilitated faster diagnostics. When the response moved on to long-term management in April 2018, Ostropaccinia psittaci had spread to most of the areas of North Island and also to the northern part of South Island. And as you know, some further spread has since occurred in the south, as well as on the North Island. Even though the response is now finished and our primary role as diagnostic service provider for the exotic organisms has been completed, we still continue to provide testing if there is a need to confirm new host or distribution records or any other really significant find that need confirmation. And we continue to provide advice as well to other MPI teams or externally. So we do keep an eye also on the new findings on science that's coming up, just to stay on the loop as well. If you want to know more about the data presented today and some additional information, this all has been published early last year in Australasian Plant Pathology. I believe Renee can also send a link out after this presentation. And it's open access, so anyone can easily get hold of it. And as I mentioned in the beginning, it is a teamwork. I'd like to acknowledge the whole Mycology and Bacteriology team who spent hundreds of hours, probably thousands, on the Myrtle Rust response. Also numerous people at MPI in the response team, in long-term management team, who we worked very closely with. Cyan carried out the host identification, assure quality, played a key role in surveillance. Department of Conservation as well had valuable contributions from natural settings. I'd also like to thank Fabi in South Africa for confirming the biotype as pandemic. And researchers from land care research, plant and food research, for their various contributions throughout the response as well as towards long-term management. And I would also like to acknowledge the contribution of all the people out there looking out for Myrtle Rust, either in the nurseries or in the communities, and helping out in other various ways. There were lots of people involved. And to finish, I would also like to highlight the care that people of New Zealand have for their plants and the environment. I'm not sure if anywhere else in the world there would be a whole page cartoon about a rust fungus published in a daily newspaper, or a website created to focus on a single pathogen in a particular country. I personally find it very impressive. So thank you all. Kia ora.
Thank you very much, Merje. Just, here we go. Get my webcam back on. Excellent. Yeah, that cartoon looks quite something. Okay, so we have one question in at the moment. I would be interested to understand how FAL MPI is screening for the potential of a new strain arriving. This cannot be detected by image, but by PCR. And do you know, Merje, how are we preparing for this?
Currently, we don't. Yes, so there has been preparedness. Considering the risk of new strains arriving here, there have been extra measures that put in place as they were for Myrtle Rust. So the ways of other strains arriving to New Zealand are very limited. And if the new strains were present in Australia, we would likely be testing to determine if that had also arrived to New Zealand through the wind. But currently we don't have any reasons to believe that there is increased risk of South African strain or any other strains of them arriving to New Zealand. But yes, there is a PCR available, yeah, which could be used.
And so that PCR test would be able to tell you it was one strain or the other?
Well, so there are methods available for it, yeah.
Okay, very good. I had one question for you. I'm not sure if you'll be able to answer this one, but just something I noticed that MPI was running awareness campaigns outside of the Myrtle Rust season, telling people to get out and look when, in fact, they're probably not going to find anything. I just wonder what the thinking was behind that. Was it that we weren't sure when we would see the symptoms in New Zealand, or was the thought that top of mind for people was so important that the campaigns ought to be run anyway?
You're talking about the late August campaign?
Yeah.
Yeah, I think I'm not responsible for that part, so I can't really reply in certain, but I think, yeah, one of the reasons was to start reminding people that it could be out there. And we did get infected material at that time as well. So we expected it to be maybe reoccurring in September. So, which, yeah, it did. So, I mean, there was a much more significant increase in November in the finds that were detected. But yeah, the idea was to have people looking, keep it in the back of their minds, and yeah, just to find any new areas or new findings as soon as possible. Because as you know, now these days there are infections throughout the year.
Okay, so we do have two more questions here, but we are at 11:30. I think I will try and get through these two questions, and people feel free to send others in, and I can get answers for you later. So thank you for your presentation, Merje. And last week, Heritage posted about the first known case of mature tree death due to Myrtle Rust that was out on the East Cape. MPI has mapped the areas where the pathogen has been detected. Do you know what the current situation is and what the most affected areas are? I suspect it's not part of your role to know that. And that's perhaps something other members of the Beyond Myrtle Rust team and By Heritage could speak to, but would you like to make a comment?
Yeah, I think so. We don't actively complete surveys anymore since it moved to long-term management. So we only get information that's sent through to us by public or survey teams. We have noticed lots more notifications from the Auckland region this summer, but not so much from the other areas. But as mentioned, if it is a new find, so we did get a new distribution record further east from Wellington a couple of weeks ago. So if it is a new finding, it does come through to us, and we record that data. But yeah,
Very
we
good.
don't have information about the significance of it. We don't measure it at the moment.
And lastly, do you have any thoughts as to how far south this disease might spread?
I think some of the work that Landcare has done is really nice, and Plant and Food as well, with the modeling, with the climate estimates, with the host availability. All them show that it could go a little bit further south, yeah, but hopefully not all the way. So we still have some refugees for the hosts that are more severely impacted.
Yes, and certainly going to depend a lot on climate in future years, as well as weather patterns within seasons within the different regions. Excellent. Well, that's it for today, and thank you very much again. A video of today's webinar will be available on the Beyond Myrtle Rust website in a couple of days, and we will send the link to everybody that registered for this webinar today. Now, the next webinar will be Wednesday, March the 10th, same time, same place, and we'll be hearing from Dr. Julia Sawato. She is currently a postdoctoral researcher at Scion. She has a long history of researching myrtle rust and in fact did her PhD on the topic. She looked at several aspects of myrtle rust in New Caledonia. Now, her talk is titled Ostropyxina psittae, the Threat of Other Strains. This will review results from New Zealand host susceptibility testing with the South African strain of myrtle rust, reminding us of the threat posed to our Myrtaceae from strains not yet here. In other words, you may think things are bad now, but they can get even worse. With that cheerful thought, I will bid you all farewell and see you back here next month.